So let's begin with another session for the day on the topic validation of analytical procedure as per ICH Q2b guidelines. We have with us an eminent speaker, Mr. Ramesh Bhusal, sir. Mr. Ramesh Bhusal, sir, is an assistant professor in Department of Pharmaceutical Chemistry. Sir has pursued his B Pharm and M Pharm from the University of Pune. He is having 1.5 years of industrial experience and 8 years of teaching experience. He has published 11 research and review papers to various national and international journals of repute. Now I would like to request Sir to take over the session.
Hello, am I audible?
Yes, sir.
Doing student, in our today's session, I am going to discuss about the analytical method validation as per the ICH guideline. Okay, now it is coded as Q2R1. Now in initial days, that was coded as a Q2b. Okay, now it is coded as a Q2R1. Okay, then that ICH word stands for International Council for Harmonization. That one is the one regulating authority, and that authority gives us the guideline about the validation of our analytical method. That one is used for the quality control tests of different type of the dosage form. Now that term is introduced by the two FDA officer. That one is a Bayer and Bird. Okay, in order to improve the quality of pharmaceutical, because they are facing the problem. That one is the sterility of large volume parenteral, and in order to sort out that problem, they are invented that term. That one is a validation. Then we have to see the definition of validation. Now there are two definitions. First one as per the WHO guideline, and the second one is as per our US FDA guideline. As per the WHO guideline, documented means our validation is a documented proof, or that one is a documented evidence. Okay, which gives us the assurance about that particular process is performed as per our requirement. Okay, that one is the definition of validation as per the WHO. And as per the US FDA, it is a documented proof which provides assurance about the particular process. If you are using a particular process for the manufacturing of particular product or for the analysis of particular dosage form, then whatever result or whatever product we are getting, that one meet its predetermined specification as well as quality attributes. Means in short, it is a documented proof that gives us the assurance about the particular process. If you are using that process, whatever result we are getting, that one is a quality result. Okay. Now, the next one is the importance of validation. First importance, that one is the reduction of quality cost. Now that reduction of the quality cost is divided into two parts of the two segments. First one is a internal failure cost, and the second one is a external failure cost. Okay. Now imagine if you are manufacturing the particular dosage form, for an example, tablet. Okay. And during that manufacturing process, if you are performing the in-process quality control test, and during that process, if our product not meeting the predetermined specification or the quality attributes, okay, then that type of the failure is called as a internal failure. Okay, means product inside our premises and it will not meet its predetermined specification or the quality attributes. Okay, then in that case, again we have to retest that particular dosage form, then we have to reprocess it or the rework it. Okay, if it will meet the predetermined specification, then we can goes for the further manufacturing. Otherwise, we have to discard that product. Okay, That came under the internal failure cost. Then the second one is the external failure cost. Now imagine if we are selling that product or sold out that product in a market, okay, and there is a complaint from the customer, okay, due to a quality problem. Then in that case, we have to recall that product and then we again retest that product, okay, and that one is came under the external failure cost. Then the second importance of the validation is a process optimization. Means if we are validating the particular process or particular analytical method, then we have to also optimize it. As the process is optimized, then we have to save our energy as well as we have to save our money. For an example, if you are set the mixing time for the mixture and that mixing time is optimized, then we have to set that mixing time, and within that mixing time we are getting a proper mixing, okay. Means by that way we have to save our time as well as we have to save our energy as well as money. And the third one, or the third importance of the validation, is a assurance of the quality, okay. Means if we are validating the particular process, then we are having a 100% assurance whatever product we are getting, that one is a quality product. Only the end product testing without validated process, it is not used as the 100% assurance, because there is a certain loopholes in the end product testing. For an example, we are testing only limited number of the sample, then the sensitivity of that particular process is a lesser one, and that's why we cannot rely on the end product testing. Along with the end product testing, if we are using a validated manufacturing process or the analytical process, then we are getting the good quality product and we are having a 100% assurance. And the last importance of that validation is a safety. Now, if we are validating the particular process, then we can assure about the safety. If you are using a autoclave, okay, and in that autoclave, pressure gauge as well as if your temperature control system is a calibrated one, then we are having a 100% assurance, or we can rely on that reading. What your reading we are getting, that one is a reliable result, and that's why it will provide the 100% safety. Now, next one is the scope of validation. Now, validation is a term that one is having a wide scope. We can validate the analytical test method, then we can calibrate the instrument. If we are using a raw material or the packaging material for the manufacturing of particular dosage form, then for its testing, whatever process we are using, we can validate it. Along with what your equipment or whatever facilities or the manufacturing operation we are using, that one is a validated one. Along with whatever cleaning process we are using, that should be a validated one. Along with if we are recruiting the operator for operating the particular equipment, then we have to check its skill as well as qualification as per our validation. The next one, validation of analytical procedure. Okay, what do you mean by the analytical procedure? Analytical procedure is nothing but the step-by-step necessary step in order to perform the analysis. Now, next one, that one is a method validation. Now, what do you mean by the method validation? Method validation is nothing but that one is a particular process, or that one is the analytical procedure used for the specific testing for the dosage form. Then next one is a parameter for analytical method validation as per the ICH guideline. Okay. Then first parameter, that one is the accuracy. Then next one is precision, repeatability, intermediate precision, then specificity, limit of detection, limit of quantitation, then the linearity study, then we have to find out the range, then we have to perform the ruggedness testing or the robustness testing for that particular analytical method. Okay. Now first parameter as per the ICH guideline is a specificity. Now specificity means we can develop such an analytical procedure which can only sense our analyte of the interest in the presence of other component of the dosage form. For an example, we can take one example. We can develop the analytical procedure as well as we have to validate it for the estimation of metformin in the tablet dosage form by using our UV visible spectrophotometer. Okay, means we can analyze our metformin in the tablet dosage form with the help of UV, and we have to validate that method. Then how to perform the specificity testing? For that purpose, in our UV visible spectrophotometer, there is a one mode. That mode is called as a spectrum mode. In that spectrum mode, we can take the Orle spectra. We can take the Orle spectra of our pure drug of metformin and the tablet solution or the our sample solution of our metformin. Okay, if we are getting the Orle spectra, means for an example, that red color wala spectra is nothing but our API wala spectra or the pure drug wala spectra, and the blue color wala spectra. Blue color wala spectra is nothing but our tablet dosage form wala spectra. If both these spectra is exactly overlap with each other, then we can say whatever analytical procedure we are developed, that one is a specific one. It will only sense the our analyte of the interest in the presence of other component of the dosage form. That may be a diluent, binder, or the glidant. Then second parameter, as per the ICH guideline, that one is a linearity study. Okay. Now, in that linearity study, we have to prepare the series of solution. In that linearity study, we have to prepare the series of solution. For an example, we are taking the metformin, pure drug, and we have to prepare the series of solution: 2 ppm, 4 ppm, 6 ppm, 8 ppm, and 10 ppm solution. Okay. And as per the ICH guideline, we have to analyze minimum 5 concentration. For that purpose, we have to take the solution from 2 ppm to 10 ppm. And then with the help of our UV, we can measure its absorbance or we can measure its response. And then that absorbance or that response is plotted against our concentration. And then how we can say that specific method is linear one? For that purpose, we can calculate the R square value. R square value is called as a correlation coefficient value. Means there is a one formula, or we are using a statistical approach, and with the help of that statistical approach, we can calculate our R square value. And if that R square value is near about 1, or it is near about 0.999, then we can say our developed analytical method is a linear one. Means whatever solution we are preparing, that one gives us the linear response. For an example, concentration of solution is 2 ppm, then its response or the absorbance is 2, and the concentration of your next solution is a 10 ppm, then we can expect the absorbance of that solution is a 10:1. Means as the concentration is linearly increases, we are getting a linear increase in the response, and that one is a check with the help of your R square value. Then the second one is a range. Okay, means we can fix the range of analysis, and whatever analysis we can performing now, we can perform that analysis within that particular range. And how we can select the range? That range is selected based on our linearity study. For an example, over here we are preparing the metformin solution from 2 ppm to 10 ppm now. Then 2 ppm is the lowest range, and the 10 ppm is a highest range. Means whatever analysis we are performing now, we have to perform into that particular range from 2 ppm to 10 ppm only. If you are performing the analysis within that particular range, then we are getting the result, or we are getting the outcomes. That one is having a precise one, that one is a accurate, as well as that one is a linear one. And the next one is a accuracy. As per the ICH guideline, accuracy study is performed with the help of recovery. Okay, now over here there is a 3 diagram. First diagram, okay, you can imagine, for an example, that one is a centralized part. Centralized part is nothing but our reference result. Yellow color wala portion is our reference result, and then that red color wala portion, blue color wala portion, or the black color wala portion is nothing but our sample ka result. Okay, and as per the ICH guideline, what is meant by the accuracy? It is nothing but the closeliness between our sample result and our reference result. Okay, which one is our reference result? That one is a yellow color wala portion is our reference result. In the first diagram, we can say there is a closeliness between our sample ka result and as well as our reference ka result. Then we can say that particular method is a accurate one as well as precise one. Means we are getting the repetitive result. Accurate means closeliness between our test result and the sample result, and the precise result means repeatability of the result. Means over here we are getting the repeated result as well as that result is closer with our reference value. Means first diagram shows our result is accurate as well as precise one. Now in second diagram, we are getting the repetitive result. Means our method is a precise one, but it is not accurate. And in the third diagram, we are not getting the repetitive result as well as what your test method we are developed, that one is not accurate as well as not precise one. Then how to perform that accuracy study? With the help of recovery. Okay, then that one is a chart for recovery study. As per the ICH guideline, we have to perform the recovery study at the 3 different levels. Means at 80% level, 100% level, and the 120% level. And at each level, we can prepare our solution into a triplicate. For an example, that one is a 1A, 1B, 1C. That one is a 3 volumetric flask. In that 3 volumetric flask, we have to add our test solution. That one is a 6 ppm, and 6 ppm ka 80%. 6 ppm ki 80% is a 4.8. Means in that 3 volumetric flask, we can add 6 ppm of our test solution or the tablet wala solution. And then in that 6 ppm wala tablet wala solution or the sample wala solution, we can add or we can spike our pure drug. How much? 80%. Means 6 ka 80% is a 4.8 ppm. Then in each volumetric flask, we can add 4.8 ppm of our pure drug. And then we can measure the absorbance due to only test solution that absorbs. For an example, that absorbance is a 0.2. And then we can measure the absorbance of our test solution after the spiking of our pure drug. Means that one is the absorbance of 0.5 is the absorbance of our 6 ppm wala test solution along with 4.8 ppm wala our pure drug. Okay, that absorbance is a 0.5. Then we can find out the recover absorbance. How we can find out the recover absorbance? From that total absorbance. Means from that 0.5, we can subtract 0.2. That one is the absorbance of our test solution. Then we are getting the recover absorbance. That one is a 0.236. And with the help of that recover absorbance, we can find out the recovered amount of our pure drug. Okay, means we are adding a 4.8 ppm of our pure drug. And after the recovery study, how much test solution or how much pure drug is recovered? That one is a 4.7 ppm. And then we have to calculate the percentage recovery, and percentage recovery for that particular test solution is a 98%. In the same order, we can prepare the 100% level wala solution, 120% wala solution into a triplicate, and we can calculate the percentage recovery. If that percentage recovery value is near about the 100%, then we can say our test method or the developed analytical method is a accurate one. Then the next one is a precision study. Now precision study means over here we can study the repeatability of the result or the reproducibility of the result. For that purpose, that precision study is divided into a 3 major types. First one is a repeatability study, then second one is the intraday precision, and the third one is a inter-day precision. Okay, first we have to see the repeatability study. For an example, we can perform the repeatability study for the 4 ppm solution, and as per the ICH guideline, we have to take the 6 solution. Okay, means over here we are taking the 6 sample solution. That one is a 4 ppm, 4 ppm, and then we have to record its absorbance with the help of our UV visible spectrophotometer. If we are getting the absorbance of that 4 ppm solution is a 0.3, 0.3, 0.3. Okay, then we have to calculate its standard deviation, and after that standard deviation, we can also calculate the relative standard deviation. And if that relative standard deviation value is less than 2, then we can say whatever method we are developing, that one is a precise one. Okay, now means we are taking the 6 solution, we are measuring its absorbance, and then we are calculating the percentage RSD. Means percentage RSD is less than 2, means it will indicate what your result we are getting. That one is a repetitive one. You can also observe that absorbance of your first solution is a 0.3, 0.3, 0.312, and the last solution is a 0.310. Means that one is a repetitive result. Then the second one, that one is a intraday precision. How to study that intraday precision? For that purpose, again we have to prepare the 6 solution having a concentration 4 ppm, 4 ppm. Okay, and then we can measure the absorbance of the solution within the same day, but at the different time interval. Means initially we have to prepare the solution, then we within a few minutes we have to measure its absorbance, then after the 1 hour, after the 2 hour, after the 3 hour, and then we can calculate the percentage RSD value. Now if we are getting the percentage RSD value is less than 2, then we can say our method is precise one. And the last one is a intraday precision, sorry, interday precision. For that purpose, again we have to prepare the 6 solution having a concentration 4 ppm, 4 ppm, and then we can analyze that solution at the day 1, day 2, day 3. For an example, if you are preparing the solution today, then we have to record its absorbance. Then tomorrow again we are using the same solution, then we have to record its absorbance, and again day after tomorrow again we have to record its absorbance, and then again we have to calculate the percentage RSD value for 0, 1 day, 2 day, and the 3 day. If you are getting the percentage RSD value again less than 2, then it will indicate what your method we are developed, that one is a precise one, or it will give us the repetitive result. Then next ICH parameter, that one is a limit of detection. Okay, as per the ICH guideline, there are 2 to 3 approaches. With the help of that, we can find out the limit of detection. Then what do you mean by the limit of detection? Limit of detection is nothing but the lowest amount of the analyte present in the sample, which is to be find out, okay, or which is to be a sense. Okay, that one is called as a limit of detection. Then how to perform that limit of detection study? By using a visual evaluation. Okay, for the non-instrumental method, for an example, we are using a titrimetric method. Then visually we can find out the lowest amount of analyte, that one is detected by the color chain. Or the second approach, we are using a signal to noise ratio approach. Okay, with the help of that approach, we can find out the limit of detection or the capacity of our method to sense the lowest amount of analyte. Okay, in that signal to noise ratio, that approach is applicable for only analytical procedure which shows the baseline noise. Okay, means it is specifically applied for our high performance liquid chromatographic studies, which shows the baseline noise. Okay, then how to find out the LOD value or the limit of detection value with the help of that signal to noise approach? For that purpose, we can find out the lowest concentration. We can find out the lowest concentration with the help of, we can get signal to noise ratio 3:1. For the better understanding, okay, now that one is a diagram. With the help of that diagram, we can easily understand what is meant by the limit of detection and what is meant by the limit of quantitation. Now you can imagine we are developing one HPLC method, and that one is a baseline noise wala peak. Okay, with the help of cursor, now that one is a baseline noise wala peak, and the baseline noise wala peak ka height is a 1 centimeter. You can imagine that one is a baseline noise wala peak, and the baseline noise wala peak ka height is a 1 centimeter. Then we are preparing the different concentration of our metformin ka solution. For an example, we can prepare the 1 ppm of metformin ka solution, and then we can inject into a HPLC, and then we will get one chromatogram. That one is a sample ka chromatogram. Okay, then we can measure the height of that sample ka chromatogram. That sample ka chromatogram ka height is called as a signal, and that one is called as a noise. And then we are measuring the signal to noise ratio. We are injecting the 1 ppm ka solution now. Then baseline noise peak ka height is a 1 centimeter. Along with, you can imagine, for an example, our signal ka peak or the 1 ppm solution ka metformin ka peak is also showing the 1 centimeter height. Then our signal to noise ratio is a 1:1. Then we can inject the 2 ppm wala solution. Then we can inject the 3 ppm wala solution. For an example, after injecting the 3 ppm wala solution, we are getting that peak, and that peak ka height, that one is the signal ka peak or the 3 ppm of metformin solution ka peak, and height of that peak is a 3 centimeter. Means if you are calculating the signal to noise ratio is a 3:1. Then which one is our limit of detection? That one is a 3 ppm solution is our limit of detection. Then that one is a second approach based on signal to noise ratio. We can use the third approach, that one is a calculation. For that purpose, we can use the formula. That one is a 3.3 into sigma divided by S. Okay, then that sigma is nothing but the standard deviation of response, and that S is nothing but the slope of our calibration curve. Okay, for that purpose, we have to construct the calibration graph. Okay, then after the constructing calibration graph, we can find out its slope. Okay, then we have to put the slope value over here, and then we can find out the residual standard deviation of our responses. How to find out the residual standard deviation for our responses? For that purpose, we can refer that chart and the formula. Formula for that residual standard deviation is nothing but the under root of summation of y minus y estimated concentration raised to the power 2 divided by n minus 2. Okay, now that one is a formula for residual standard deviation ka calculation. With the help of that formula, we can find out that sigma value or the residual standard deviation value. Then we can put that value over here, slope ka value over here, and then we are getting the detection limit. Okay, for an example, we are preparing the 4 solution, that 4 solution having a concentration 1 ppm metformin, 2 ppm for metformin, 3 ppm, and the 4 ppm metformin solution. Then we are recording its absorbance. Absorbance is nothing but the Y. Then 1 ppm ka solution absorbance 4, 2 ppm ka solution absorbance 4. Then next one is the 6, and the 4 ppm ka solution gives us the absorbance 7. Then we can find out the Y estimated. How we can find out the Y estimated? That one is a find out with the help of our straight line ka equation. Which one is a straight line ka equation? If you are plotting a graph of concentration versus absorbance, then we are getting one straight line ka equation. That straight line ka equation is nothing but over Y is equal to mx plus c. That Y stand for absorbance, m stand for slope, x stand for concentration, and c is nothing but the y-intercept. Okay, this over here we are already aware about the concentration, we are already aware about the slope. Then we can put the concentration ka value, slope ka value, and the y-intercept ka value. Then we are getting the y established value. Okay, for an example over here we can consider that one is a y-intercept ka value is 2 and the slope ka value is a 1. And if you are putting that entire into a formula, that one is a straight line ka formula, y is equal to mx plus c, then we are getting the estimated value. That one is the estimated absorbance. That one is a 3, 4, 5, 6. Then we can record its difference. Which one is the difference? We can take the difference. That one is a y minus y estimated value, and then we have to take its square, and then we can carry out the summation of that residual value. Okay, means we are getting ye wala value. That one is the summation of y minus y estimated ka value. Then we have to put that value over here. Then we have to put the n ka value. N ka value is nothing but our number of observation. Number of observation is a 4. Then 4 minus 2, that one is a 2. And if you are calculating that residual standard deviation value, then that residual standard deviation value is a 1.7. Then that 1.7 value we can put into our formula, means 3.3 into 1.7 divided by slope of our calibration curve is 1. Then we are getting the detection limit. Means there are 3 approaches: with the help of visual evaluation, with the help of signal to noise ratio, and with the help of calculation. We can find out the limit of detection value. Then next one is a limit of quantitation. Okay, as per the ICH guideline, next parameter is a limit of quantitation. And what do you mean by the limit of quantitation? It is nothing but the capacity of our method which can quantify the analyte present in the sample. Okay, for that purpose again we can use the different approach, same like that of our limit of detection. That one is a visual approach, signal to noise ratio approach, and that one is a formula approach. And with the help of that we can find out the limit of detection. Okay, then limit of quantitation. Then formula for limit of quantitation is equal to 10 into sigma divided by S, and that S is calculated from the calibration curve, and the sigma stand for, that one is the residual standard deviation, and with the help of that we can find out the quantitation limit ka value. Then the next approach as per the ICH guideline, that one is a ruggedness study. Now in the precision study we are checking the repeatability of the study. Repeatability of the result. Sorry, in the precision study we can check the repeatability of the result. But in that precision study, we are check the absorbance of our solution by using the same instrument, by using a same lot of reagent, or by using a same analyst. And in the ruggedness parameter, we can check the repeatability of the reproducibility of the result by using a different condition. Means we are using a same solution but condition is a different. For an example, we can measure the absorbance in different laboratory by using a different analyst, by using a different instrument, different lots of reagent, different temperature, and we can perform the analysis at the different days. Okay, if after checking the absorbance of the same solution under the variety of the condition, if we are getting the repetitive result, and how we can say we are getting the repetitive result? With the help of our percentage RSD value. Okay, we can calculate the percentage RSD value, and if that value is less than 2, then we can say our developed analytical method is a rugged one. Then the last parameter from that ICH guideline, that one is a robustness study. In that robustness study, we can change capacity of our method. That one is a remain unaffected by small but the deliberate variation in the method parameter. For an example, we are developing the UV method, then which one is a method parameter? That one is a lambda max value. For an example, initially we are using a 250 nanometer as an wavelength for the analysis of the metformin. Then in that robustness study, we can modify that parameter. Means instead of 250 nanometer, we can measure the observance of the solution at the 248 nanometer, at 250 nanometer, and at 252 nanometer. Means we are varying the parameter and then we are checking the effect on our result. Okay, if still we are getting the repetitive result, then we are getting whatever analytical method we can develop, that one is a robust one. Thank you, student, for your patient listening. I'm also thankful to SCIENTIA organizing committee, Mr. Tanvir sir and the Trupti ma'am. Along with, I'm also thankful to our respected principal ma'am, Dr. Savita Tauro, who gave the opportunity to deliver that presentation under the SCIENTIA. Thank you. Now the session is open for question and answer. If you are having any query or the question, you can ask me.
Good afternoon, everyone. I would like to take this privilege to propose a vote of thanks to our eminent speaker, Mr. Ramesh Bhusal, sir, for his informative session on validation of analytical procedure as per ICH Q2b guidelines. Sir has focused on the importance of validation, the scope of validation. He has described about various about method validation and various ICH parameters of analytical validation. Our sincere thanks to Chairman Mr. Albert D'Souza sir for his encouragement, and thanks to all other dignitaries of the management. A special thanks to our principal, Dr. Savita Tauro ma'am, for her immense support. I would also like to thank the coordinators for their guidance and support. The feedback link will be provided in your respective WhatsApp group. Kindly fill it, as it will help us to improve our program. And last, thank you for being there with us. Have a great day.